All right, folks. So we're going to do chapter 12 here. Some of the material that I have talked about in lecture. So we'll just kind of quickly move through that here. Let's quickly review what the nervous system is. That's going to be depending on, well, we're going to be going from it from an anatomical standpoint. So the nervous system is going to be made up of the central nervous system and the peripheral nervous system. So our central nervous system is made up of our brain and spinal cord, and our peripheral nervous system is going to be made up of nerves and ganglia. And so we defined nerves before as bundles of parallel axons, all right, in the peripheral nervous system. And ganglia were just a cluster or grouping of cell bodies. So our nervous system is going to be, okay, one of two communications control systems of the body. It plays a primary role for control. The endocrine system is involved in that also, but it's going to be communication and control. And so it does all of its functions, all right, through electrical activity. And so we have our neurons that are going, and which we'll talk about when we talk about a little bit in lecture. We're going to see how, all right, these neurons are going to utilize electrical activity to integrate and regulate the functions of our body. And it does so using that homeostatic mechanism, all right, the functions of collecting information, processing and evaluating that information, then initiating that response to that information through the effectors. Okay, so quick review here. A nerve is a bundle of parallel axons in the peripheral nervous system. A nerve will be covered by three coverings: the epineurium, the perineurium, and the endoneurium. The outermost layer is going to be the epineurium that covers the entirety of the nerve. Right? Its role is going to be support and protection, and partly because that comes from that dense irregular connective tissue. Right? Then we'll see that some of these axons, all right, we've bundled together into these structures called fascicles, and they're just a bundle of axons. And then we'll wrap that up again in another dense connective tissue, that dense irregular connective tissue, and that will be an inner layer. And then the innermost, which wraps each individual axon, is the endoneurium. That's that loose connective tissue there, and it helps to physically separate each axon from one another, but it also will insulate those axons, so our electric charge doesn't jump around. Right? And then, of course, nerves are highly vascularized, and we'll see various blood vessels throughout the different layers there. All right, so here's that picture that I showed you earlier. All right, here's our epineurium, and then our perineurium around the different fascicles here. And then you have your endoneurium that is going to be covering each individual axon there. And then you can see down in here is our ganglion. All right, this is the clustering of those cell bodies, right, in the peripheral nervous system there. So a quick review on classification of our nerves and ganglia. Okay, keep in mind when we're talking about anatomically, right, if a nerve comes off the brain, it's a cranial nerve. If it comes off the spinal cord, it is a spinal nerve. That means cranial nerves and spinal nerves are both peripheral nerves. Now, when we discuss it from the functional or physiological classification, we're going to talk about what these nerves do. Sensory nerves contain only sensory neurons, and they're going to send information to the central nervous system. That's sensory input. Motor nerves only contain motor neurons, and that is going to send information from the central nervous system out to the effectors, okay? And so that is going to be made up only of motor neurons. And then mixed nerves, like the name implies, it's going to have both sensory and motor neurons. And so keep in mind, all right, those axons for those individual neurons are only going to carry one specific type of information. All right, so a ganglion is a cluster of neuron cell bodies in the peripheral nervous system there. Okay, ganglion is a cluster of neuron cell bodies in the peripheral nervous system. All right, so 2 types of cells in nervous tissue, the neurons, the rock star, okay, the diva, that is going to be the cell that when we're talking about the different characteristics of nervous tissue, all right, when we talk about excitability and conductivity, that's the cell that we're talking about. That's the neuron. These are those cells that when they become excited will transmit electrical impulses or signals. Then, of course, there's the glial cells. They're the non-excitable cells. Okay, they cannot transmit information. But what is their job? Everything else. Okay, they are the gopher. They are the protector. Okay, they will support and protect the neurons and do so much more, and which we'll talk about here when we go over the different types of cell types here. Okay, here are those characteristics again that I showed you before: excitability, conductivity, secretion, extreme longevity, and amitotic. Right? Once, all right, the neuron reacts to whatever that stimulus is, all right, we have to grade it, and we use excitability to characterize that. Its responsiveness to a specific stimulus. And we didn't really quite get into it completely, but when we talk about that cell's membrane potential, what we're talking about is that electrical charge across the plasma membrane. So basically how we can change that electrical charge, all right, will determine its excitability. Then once we've actually generated that action potential, that electrical signal, then we have to keep it alive. And we got to get it from point A all the way down to point B. Right, so that when we talk about the conductivity, we're talking about the ability to propagate that electrical signal there, right? And so we'll discuss that. And we already now know what these voltage gated channels are going to do. So normally those voltage gated channels are closed, so nothing will be happening. But once we have the proper type of stimulus, we will open up those voltage gated channels, okay? And then we'll see how they actually will conduct that electrical signal down the entire length of the axon there. Once we get to the end of the axon, we'll get to the transmissive segment of our neuron where the synaptic knobs are. And inside the synaptic knobs, we have those synaptic vesicles which contain the neurotransmitter. Yes, our beloved neurotransmitter, that chemical that our neuron is going to release onto whatever the target cell is. So it could either be a muscle, a gland cell, or it could be another neuron. Okay, talk about how our nerves, the neurons, pretty much live forever, or pretty much the lifetime of the person, and then they do not divide after fetal development. All right, parts of the neuron real quick. Okay, the cell body, I did not mention this before, but the cell body is not myelinated. What does that mean? Myelin is this fatty tissue, okay? When I say fatty, it just has a high composition of lipid in it, okay? And so it appears if you have myelin, which is this lipid-rich substance that surrounds these nerves, okay, it appears white. So if it is not myelinated, it will appear as gray matter. So you'll hear the term white matter and gray matter when we're talking about the central nervous system, okay? So if you're gray matter, that means you're not myelinated, okay? Because myelin only is going to appear in certain areas, and we'll talk about that today. The cell body is one of those areas where there's not going to be any myelin. So that will appear as gray matter. So when we're talking about the plasma membrane of the cell body, okay, we refer to that, all right, the plasma membrane, all right, for the cell body is the neurolemma. And then inside where the cytoplasm is, we have a new name for that. That is the perikaryon. Right, and I mentioned this before, how the chromatophilic substance that we call Nissl bodies is going to be found here in the cell body there. And that's just basically ribosomes, both the free and bound ribosomes. And then we'll have these short processes that are coming off of the cell body. and those are called dendrites, and they're also not myelinated. They're not myelinated, so they'll appear gray. Okay, and so what is their role? Their role is to receive input. Okay, so they'll receive the input and then they'll try to transfer that input information, graded potentials, onto the cell body or the soma there. Okay, the axon, I didn't mention this in lecture, does not have chromatophilic substance. That's only going to be found in the cell body. All right, so that's that longest and biggest process that comes off of the cell body, and it's going to run all the way from the cell body down to either the target cell or another neuron. So it's going to contact either another neuron or one of our effectors. So where the axon attaches onto the cell body, we call that the axon hillock. That is that triangular region there. That's where we're going to initiate our action potential. And then inside there, of course, we're going to rename the cytoplasm in the axon. It's called axoplasm. Cell membrane is called the axolemma. Sometimes our axons will have branches that come off. They'll go a bunch of different directions. And those are called axon collaterals. They might go out to another muscle somewhere else or another gland somewhere else. Right, and at the very end of our axon, we have these little branches that come off, and those are our telodendra, the axon terminals. That's where the axon ends. And at the end of this telodendra, we have these swellings. All right, those are our synaptic knobs. And inside are the synaptic vesicles that contain the neurotransmitter, okay, which will be released. Once an action potential comes down the axon, it'll stimulate the release of that neurotransmitter, all right, from the synaptic vesicles, and it'll diffuse across the synaptic cleft to the other side of the synapse there and hopefully bind on to a chemically gated ion channel. All right, I already talked about the cytoskeleton there. And here you can see this picture should look familiar to you, all right, and how you can see, okay, we have a couple neurons here just to put it into perspective. Here's our cell body. You'll notice there's no myelin. These cells here, these neurolemmacytes, they make the myelin. So they are not going to be present on cell bodies or dendrites. So areas that have a lot of gray matter, we'll have a lot of dendrites there, or we'll have a lot of cell bodies in that area. And of course, the dendrites are part of the cell body. So you can see we have our long process here. That's going to be the axon. And you can see the space in between the cell body and the axon. That's the axon hillock. All right. And then you can see here's an axon collateral. And at the end of our axon, we have our telodendra. Those are those terminal extensions that end here at the synaptic knobs there. Okay, so now let's go and talk about some new information here and how we classify our neurons. And so how we classify the neurons structurally is dependent upon the number of processes that are coming off of the cell body. Okay, the number of processes. So here you can see we have multipolar neurons, bipolar neurons, unipolar neurons, and axonic neurons. So the one that we've been talking about the most is the multipolar neurons. That's the most common type. So when you say multipolar, that means off of the cell body, you're going to have many dendrites and one axon. That's that picture right there. Okay, that's an example of a polar neuron. Many dendrites and one axon. Okay, and so we're going to see this, you should know this, in all skeletal muscle motor neurons. All skeletal muscle motor neurons. So going back now, I'm going to pull a little bit of lecture here back on you. Remember what I said when we were talking about the different, how we classify the nervous system, when we're talking about how we classify it functionally. Okay, you've got your sensory and motor. And then in the motor nervous system, or the efferent nervous system, you had the somatic motor nervous system. Then you have the autonomic motor nervous system. Well, remember what we said about the somatic? The somatic motor nervous system, all right, is what you can voluntarily or consciously control, the effector. And the only type of tissue that you can voluntarily control is skeletal tissue, skeletal muscle tissue. So therefore, all the skeletal muscle tissue motor neurons are going to be multipolar. So you can also say that all the neurons in the somatic nervous system are multipolar. All right, so the second type of neuron is the bipolar. And so you have one dendrite coming off of one end of the neuron, and then off the other end, you have the axon. So you're going to see this, and we'll see this in chapter 16, all right, when we talk about the eye. So we'll see those in the retina. Then you have your unipolar neurons, all right? So you have one process that's coming off of the cell body. And then what happens is that process then splits like a T, one going one way, the other process going the other way. And so it splits into two processes. So most of the sensory neurons that you're going to see, all right, in our periphery are going to be unipolar neurons or pseudo-unipolar. So one process is the peripheral process, and that's basically going to have like the dendrites on it. And then the other process, the central process, that end is going to have the synaptic knobs. Okay. That's a horrible drawing. And that's going to head towards the central nervous system. And then finally, we have the anaxonic neurons, and they have dendrites, but no axons. So if you don't have an axon, you can't generate action potentials. So these types of neurons only can generate graded potentials. We'll learn more about those later on. Not today, though. All right, so here's our picture of what these neurons look like here. All right, so here's the multipolar. Okay, so here's the cell body. You see all these dendrites, and you have one big long process. That's the axon. That'll transmit the nerve signal. Then we have our bipolar. So again, our cell body's in the middle. One process off of one end is going to have the dendrite. The other process is going to have the axon. Then we have our unipolar neuron. Here's our one cell body with the one process coming off. Then it splits into the peripheral and the central processes. So one end is the dendrites. This is going to be down by where the receptors are. And then over here, the central process, that's where our synaptic knobs are going to be, and that's going to head towards the central nervous system. And then finally, our anaxonic neuron. Okay, so you have your cell body and then all these dendrites. Okay, but since it does not have an axon, it cannot generate action potentials. Don't be fooled. All right, so that's how we classify the neurons structurally based on the number of processes coming off the cell body. Now let's classify our neurons functionally, okay? And it's important that you know this, all right? We classify it in the direction that the nerve signals or action potentials are moving, okay? So real simple. Sensory neurons, also known as afferents, they're going to conduct sensory input information to the central nervous system. So that means it's going to conduct input from either, well, I shouldn't say either, from the somatic sensory nervous system and the visceral sensory nervous system. It's going to conduct that input from those receptors to the central nervous system. Right, so again, we're going to see, all right, most of these are unipolar. Well, what does that look like? In case you forgot, that's this fella right here. That's the unipolar one. So we're going to see the cell bodies in the posterior root ganglia. I don't know if I have a picture for that. Oh, I do. Okay. Here's the posterior root ganglia, also known as the dorsal root ganglia. Thought I had another picture. Yeah, here we are. So here is what we're looking at. We're just going to zoom in here, so you can see all these cell bodies right there. Those are unipolar, and that is the dorsal root or posterior root ganglia. Okay, then when we're talking about motor neurons, the efferent, the effectors, they're going to conduct that motor output from the central nervous system out to the effectors, right? Either to the somatic effectors or the visceral effectors. Right? So we talked about this before, all are multipolar. Here's though an important fact though. Most of the cell bodies will be located either in the spinal cord or your brain. Right? They'll be located in the central nervous system. And then we have the interneurons. These neurons are cool because not only do they receive information and process it, they're going to integrate that information and kind of determine what is going to happen. They have the power, all right, to stop things dead in their tracks or to start things up and continue certain processes, okay? So interneurons do a lot. And so it's important to understand that it's these neurons that are found in between the sensory and motor neurons. So they're going to allow the sensory input aspect of your nervous system to communicate with the motor output, all right, or the effector portion, okay? And guess what? 99%, it's a cool number, all right? 99% of your neurons are interneurons. That means 1% of the neurons in your body are sensory and motor neurons. Pretty crazy, okay? And so you'll find the interneurons located only in the central nervous system. So this picture here really, and I hope that you get a good understanding from this picture, right? When we're talking about how the functional aspect of the nervous system works. Basically, all right, we have receptors in the skin for this example. Okay, so you have skin receptors, which are going to then trigger or be set off by a stimulus, and they're going to transmit that sensory input or that sensory information along a sensory neuron. Sensory neurons carry information from the periphery, from the receptors to the central nervous system. So this sensory input will travel on our sensory neuron. The cell body of that sensory neuron lives here in the dorsal root ganglion or the posterior root ganglion. It'll enter into the spinal cord here and it'll synapse onto this fella here, the interneuron. And so this interneuron then is going to kind of link the sensory part to the motor part. And so that interneuron will then transmit information here to the motor neuron, and it'll stimulate the motor neuron then to send that motor output information from the central nervous system down to the effector. And in this case, it's the skeletal muscle. So this is what we're going to talk about in later chapters. First, we're going to learn how these neurons work, how we turn them on, how we turn them off. Okay, but then when we get into the later chapters here, when we're talking about the nervous system, you need to understand this kind of pathway here, okay? All right, so we talked about the neurons. That's one cell in our nervous system. Let's talk about the glial cells, also known as neuroglial. These are the ones that are the support cells. They offer protection. They're the non-excitable cells, okay? So their job, you're going to see them throughout the central nervous system and also in the peripheral nervous system. Here's the crazy part, okay? Yes, they're smaller and much smaller, okay? But they make up volume-wise half of the nervous tissue. So that means even though they're smaller, right, they are going to outnumber the neurons because they make up half the volume. These cells can divide. And these cells all right, cannot transmit impulses. So they're like the police, they serve and protect, okay? So they're going to protect the neurons, they're going to provide, all right, nutrients for the neurons. If the neurons get damaged, they'll help to repair that damaged tissue. Glial cells during development help to kind of lead the neurons, all right, as they're growing, helps to lead the neuron, all right, to where it needs to go. And, all right, we don't appreciate it very much, but they play a huge role at the synapses. They're the cleanup crew. I don't know if anyone here has ever been to New York City or Times Square on New Year's Eve. I've been, and it's amazing because the very next day, if you're walking around Times Square, you'd never know that there was one of the biggest parties on the planet there, because they clean everything up. They do such a wonderful job. And I mean, as soon as that ball drops and people start filtering out of that place, there's people out there cleaning. And that's what the glial cells do. The glial cells at those synapses, remember what I told you, that neurotransmitter spills out into that synaptic cleft there. Well, it just doesn't just hang out there. There's several things that will occur, right, to those neurotransmitter molecules. But eventually, right, we've got to shut the party down. So the glial cells will help with cleaning out those neural synapses. Pretty cool. So let's start talking about the first type of glial cell, the astrocytes. Astro, I think of star, okay, so these are star-shaped cells, and they have a bunch of processes that come off of the main body there. And at the end of those processes, they have these swellings, right, these structures called perivascular feet. And so these perivascular feet, right, when we're talking about the blood-brain barrier, the BBB, right, they will surround the capillaries there of the blood-brain barrier. And so they will allow certain substances to cross over from the blood into the nervous tissue and vice versa. So it's very helpful because, right, the blood-brain barrier will prevent certain things from entering your nervous tissue, i.e. certain pathogens, certain toxins, poisons, okay? And it'll allow the passage of certain nutrients, okay? So it's very helpful. Astrocytes play a big role in our potassium concentration, you know, within the nervous system because, and we haven't really quite talked about it, and we will as we move through chapter 12, but the concentration levels of certain electrolytes, certain molecules is crucial. Okay, potassium is one of those. If you have high levels of potassium, it can be bad for your heart and other tissues, and vice versa. If you have low levels, there can also be issues. So it's really important to maintain these stable chemical environments around our neurons, right, so they can function properly. Okay, because if some of those elements or those molecules get out of whack, it can cause certain physiological issues going on. All right, astrocytes help with structural support. I mentioned before how when the neurons are developing during that fetal development period, right, how certain glial cells will help with that. Well, the astrocytes help with that neuronal development. And what I really like is that these cells, all right, will start to occupy the space, all right, of dying neurons. I don't know why I always found that intriguing, okay, but what will happen is the neuron is degenerating, okay, these astrocytes kind of move into that space and occupy that space to help maintain some of that structural integrity of whatever that nervous tissue is part of. Ependymal cells. I'm sure you folks have heard of cerebral spinal fluid, very important substance. We'll talk about that in more detail later on. All right, but cerebral spinal fluid, right, is created by the ependymal cells. And basically, they filter out the blood and the blood plasma specifically. And then that's how they make that cerebral spinal fluid. And they do so as part of the choroid plexus. So you'll see inside your brain, you have these cavities. and those cavities will be lined by the ependymal cells, and you'll have these clumping of these ependymal cells called the choroid plexus, and they just will filter out that cerebral spinal fluid. It's kind of cool. Microglia. Okay, we need to have some immune cells around our nervous tissue to help protect it. So the microglia are like the central nervous system's version of the macrophage, okay, except these guys are small. And so they just travel around and just kind of checking everything. And of course, if they run across some sort of pathogen like a bacteria or whatnot, right, they'll initiate the immune response. Most times they'll just undergo phagocytosis and just gobble up whatever it is that they found, right? And of course, if there's an infection going on, they'll start to reproduce, to help match that infection there. If there is tissue damage up there too, they'll also act similar to the macrophage and help to remove some of the debris there. Oligodendrocytes, right? These cells, their job, right, is going to be to create myelin. And myelin is this fatty tissue, okay, that wraps around our axons of our neurons and helps to insulate the neuron. And by insulating these neurons, it allows for that action potential, that nerve signal to move faster. So myelinated neurons, all right, will be able to transmit an action potential faster than unmyelinated neurons. Okay, that's called saltatory conduction. We'll talk about that in chapter 12. Great picture here. It shows you all the different cell types. Okay, it's pretty easy to figure out where the neurons are. So you can see here the neurons. Okay, here's our astrocytes, star-shaped cell with these processes coming off. And you can see when these processes come in contact with the capillary here, right, it's going to regulate what can enter into the cell and what can leave, what can leave the capillary, right, and enter into the nervous tissue here. And so it plays a big role in that, in helping to transmit certain substances out of the blood and into the nervous tissue there. It's also, you can see here's some of our microglial cells. They're all over the place, these small little cells here, right, with their immune function. Our oligodendrocyte, you can see it's got its extensions here, and these extensions will wrap themselves around, right, parts of the axon, usually about 1 millimeter in width here. But you can see how one oligodendrocyte can myelinate several different axons. So an oligodendrocyte can myelinate more than one neuron. Right, and then over here, you can see our ependymal cells, and they're lining one of the spaces of the brain here, the ventricles, and they're going to filter out and create that cerebral spinal fluid there. I think I hit over all the cells. Yep. All right, so those were the glial cells of the central nervous system. The glial cells of the peripheral nervous system are a little bit easier because there's only two that we're going to talk about: the satellite cells and the neurolemocytes, also known as the Schwann cells. All right, think about the satellite cells. You're only going to find these folks around the cell bodies in the dorsal root ganglion or the posterior root ganglion. And so, again, being a glial cell, they're a support cell. So they're going to help with the movement of nutrients and wastes, but also they will electrically insulate those cell bodies in the ganglion. So they're going to just, you'll see it, I'll show you a picture in a second, how these satellite cells would just surround the actual cell body. And by doing so, they help to insulate it. The neurolemocytes, the Schwann cells there, these guys are going to make the myelin in the peripheral nervous system. We talked to you about, I told you what the myelin does. It helps to increase the speed of that action potential. Now, here's the difference between the neurolemocytes and the peripheral nervous system compared to the oligodendrocytes of the central nervous system. Here's our oligodendrocyte, and you can see, right, how the oligodendrocyte is myelinating several neurons. Well, in the peripheral nervous system, the neurolemocyte only can myelinate one single axon. And in fact, the cell itself will form that myelin sheath. We're going to talk about that. All right, so here you can see in our peripheral nervous system, here are those satellite cells, and they just surround the cell body. They help to insulate it, and they help to provide nutrients and get rid of wastes. Okay, and then you can see over here, here's an example of a neurolemocyte. All right, so the entire coating itself is one single cell. You can see here's the cell nucleus pushed up against the side. And what this does is it helps to electrically, all right, insulate the axon so we can increase the speed of our action potential. Okay, I'm not going to talk about that. Let's talk about the myelination process here. Okay, so our myelination process starts, okay, with, all right, well, the cell that's actually going to create the myelin. So in the peripheral nervous system, that's the neurolemocyte or the Schwann cell. In the central nervous system, that's the oligodendrocyte. Okay, but you have to understand, all right, that myelin is a lipid substance. And so that's why when we're looking at it, all right, grossly, it looks white. So if you're seeing, remember I was telling you before, gray matter and white matter, if you see white matter, then that white matter, all right, it gets that appearance from the myelin sheath that is coating, all right, or wrapping itself around the axon. And what does myelin do? It insulates the axon and makes it so that that action potential can move or propagate faster. All right, so let's talk about the peripheral nervous system. Let's talk about the Schwann cell, the neurolemocyte. So basically what happens is this cell will start to wrap itself around the axon. And as it does that, okay, it starts to push the nucleus along with the cytoplasm out to the periphery. And so all of that cytoplasm and the nucleus that's pushed out to the periphery, we call that the neurolemma. Okay, so one single neurolemocyte will actually myelinate 1 millimeter of the axon. So you need a lot. You need several neurolemocytes, okay, to line or cover the entirety, because some of those axons are pretty long. You can have an axon that can run down the entire length of your arm. Okay, so we'll need several. So if you notice with some of these axons here, okay, there are these gaps in between, all right, these neurolemocytes. See these gaps, here's one, you can see a bunch. Here's a gap, here's a gap, here's a gap, okay? And so those gaps, all right, those are called neurofibril nodes or the nodes of Ranvier. So here's that myelination process. You can see, all right, we have our neurolemocyte, and what it does is it starts to wrap itself around this individual axon. And so you can see as it starts to wrap itself around, it's going to eventually start to push that cytoplasm and the nucleus out to the periphery. Here we go. You can see as it keeps wrapping itself around, there's that nucleus, it's pushed off to the edge. Here's the cytoplasm, it's pushed off to the edge until it's finally done. And so where we see, all right, the nucleus of the Schwann cell and the cytoplasm there, all right, all that on the periphery, that's called the neurolemma. And then you can see these tightly wrapped layers around the axon itself. That's the myelin sheath. Okay, so one single cell in the peripheral nervous system, one single neurolemocyte is going to wrap itself around the axon. It's roughly about 1 millimeter in width. So here you can see all that fun stuff. Okay, so here's our neurolemocyte. Then there's that space there. That's the node of Ranvier or the neurofibril node. And then you can see the myelin sheath is that part that's covering, all right, those thin layers that are wrapped around themselves consecutively. And then the outermost part, okay, the outside of our neurolemma site, that's what we call the neurolemma. And that's going to include the Schwann cell and the cytoplasm, all that pushed up against the edge there. All right. Nope, sorry, I'm running all over the place. Okay, so keep that in mind. Our neurolemma site, right, is going to be in the peripheral nervous system, the cell that myelinates the axon. Okay. In the central nervous system, we have the oligodendrocyte. Now, remember what I said, one oligodendrocyte can myelinate multiple axons, but roughly about the same amount of myelin. When I say the same, all right, when we're talking about the dimensions, it's about 1 millimeter, okay? But it can myelinate multiple axons, and it can myelinate multiple the same axon, but in multiple spots, okay? So here's the thing, and this is important. The difference between the two, other than location, is with the oligodendrocytes, you will not have a neurolemma forming because the cell is not wrapping itself around, all right, the axon like the Schwann cell did. Okay, the oligodendrocyte just shoots this extension out at it, and it covers the axon with the myelin sheath there. Okay, but we will have those neurofibril nodes there that we saw, just like we saw in the peripheral nervous system. Okay, so when we're talking about unmyelinated axons, no myelin sheath. No myelin sheath. Okay, so what we'll see though, okay, with these unmyelinated axons, when we're talking about the peripheral nervous system, okay, those neurolemmocytes, right, they won't wrap themselves completely around these these axons like they did with a myelinated axon. All right, what we'll see instead is like this depression into the cell where the axon is kind of pressed in. Okay, but it doesn't wrap itself around like we saw before. Okay, so let me show you some pictures. Okay, so here you can see in the peripheral nervous system, all right, we have a bunch of axons. Okay, they're just together, and then you got this neurolemmocyte that comes over to it, and it kind of gobbles them all up. All right, and then the cell itself normally will just wrap around one individual one. In this case, what happens in our unmyelinated axon is, all right, our nucleus is in the center, and it pushes the axons to the periphery here. And so they're exposed to the outside here, but they haven't gotten that myelin sheath. So that's what it looks like in the peripheral nervous system. Okay. And then, oh, sorry. And then the central nervous system, you're not going to see oligodendrocytes having anything to do with the unmyelinated axons. All right, which brings me to, since we're talking about myelination, all right, multiple sclerosis and Guillain-Barré syndrome. Okay, one is going to affect the central nervous system, the other one will affect the peripheral. So when we're talking about the central nervous system and the myelination process, multiple sclerosis is when we see demyelination of the neurons in the central nervous system. And we say it's a progressive condition because it just continues on and usually it gets worse. Unfortunately, it's an autoimmune disorder for whatever reason, right? These cells start to demyelinate, okay, because of our immune system. And unfortunately, we see it in a higher number of women than we do men. So the immune cells just start to go to work on those oligodendrocytes, and it attacks. And anytime, all right, you suffer damage, one of the first things that occurs in any type of tissue damage is inflammation, swelling. And when you get inflammation, you get degeneration of whatever that tissue is. And as a result, your body tries to help and fix everything. And so you get scarring. And we talked about what happens with fibrotic tissue. Normally, you lose organ function, and that's what happens here. Okay, we now have a permanent loss, right, of that organ function because we attack those cells, damage those cells, and now we have damaged neurons as a process, as a part of that process there. Okay, so you'll see a slew of different signs and symptoms. Vision problems, muscle weakness is usually one of the earlier signs that you'll see. Some folks, depending on the age of the person, okay, with the vision problems, right? Bowel and bladder issues going on, okay, and mood problems, especially when you're dealing with some of those central nervous system neurons there that affect mood, your limbic system and whatnot. That could be a big problem there. All right. Now, if we're going to deal with the peripheral nervous system, right, we have Guillain-Barré syndrome, in which we start to see that demyelination process in the peripheral system nerves there. And of course, it's that inflammation that causes that damage. Okay. And as a result, right, you get damaged to that area with the swelling. The nice thing is that the peripheral neurons can recover from this. So initially what you'll see is, right, folks will complain of having issues with like their fingers. They'll start to drop things. They can't really control their motor function all that well with the distal limbs. And then as it progresses, all right, it'll move more towards those proximal muscles. They might have hard time bending their elbow, right, as things get more advanced. Now, Guillain-Barré, you can recover from. Okay, so most of the time, full restoration of organ function occurs if you just kind of let it run its course. We really do not have a specific idea as to what causes this, right? But usually it's some sort of infection. Okay, so if it's a viral or bacterial, right, monitor any symptoms in regards to fever and try to treat the symptoms accordingly. If it's obviously antibiotics, if it's a viral, if it's bacterial. Okay, but in most cases, people that get Guillain-Barré make a full recovery. All right, so I think no. Okay, I do want to just quickly throw this slide up here to review at this point, all right, some terms that we will be using throughout the remainder of this course when we're dealing with the nervous system. Okay, so you have white matter of the CNS, gray matter of the CNS, a nucleus, a ganglion, a tract, and a nerve. Some of this looks familiar, some of this doesn't. So I feel like this is a good time to discuss this. All right, so white matter of the central nervous system is going to be, this is the only definition. And so if you know this definition, you won't have to worry about gray matter because there's way more stuff in gray matter. So white matter is going to be myelinated axons because we just talked about the myelination process. And myelin is going to be in one place. That's the axon. It doesn't go anywhere else. So when you see white matter in the central nervous system, in the brain and spinal cord, you'll see that white matter. You'll be like, I know what that is. That's myelinated axons and nothing else. All right, now, when we get to the gray matter, it's a different story. It's going to include unmyelinated axons. Okay, that makes sense because it's the myelin that gives the tissue that white appearance because of the high lipid content. All right, so now we're looking at gray matter. So we're saying, okay, that's unmyelinated axons. But when we're looking at the actual neuron, we noticed that myelin was not on the cell body. Okay, so myelin doesn't appear on the cell body, and that includes the dendrites. So gray matter is also going to be the neuron cell bodies and then also glial cells. So when you're looking at gray matter, it's going to be unmyelinated axons, glial cells, and cell bodies. All right, so these last four terms, okay, are going to be location specific. Let me repeat that. These last four terms are location specific. If we're dealing with the central nervous system, we're going to call, all right, a specific structure one name. If we're dealing with the central nervous system, we're going to call that same specific structure a different name. Okay, so let's start off with what we know. Go all the way to the bottom here and look at nerve. A nerve is a collection of axons, parallel axons in the peripheral nervous system. Okay, we already knew that. And let's go up here to ganglion. A ganglion is a collection of neuron cell bodies in the peripheral nervous system. Okay, so now what do we call a collection of parallel axons in the central nervous system? We call that a tract. Okay, same thing, different location. So a nerve is in the peripheral nervous system. A tract is in the central nervous system. A ganglion is a collection of neuron cell bodies in the peripheral nervous system. Now we have a new term, a nucleus. Oh, wait, Dr. Kaz, isn't a nucleus the control center for a cell? Yep, that's the definition of it, all right. But we have another term, okay? Nucleus is going to be a collection of neuron cell bodies in the central nervous system. Note, it's not the same thing as the nucleus in a cell. Completely different, okay? Don't ask me why they did this. I can't remember. I knew at one point. Sometimes information just falls out of my head and I can't remember why. But at one point I knew why they called it a nucleus. Okay, but please know that terminology. It'll help you. So what happens when we injure an axon? Okay, it happens more than you'd like to care about, okay, but it does happen. All right, so when we're dealing with traumatic injuries in the peripheral nervous system, we are grateful that the axons can regenerate. And that regeneration is going to be possible, one, if the cell body is intact, if it is not harmed. Okay, and two, if we have enough neural lemma to work with so we can regrow that axon. Remember what the neural lemma was? Okay, that was that outer portion of that neural lemma site that included this, the cytoplasm, all right, the plasma membrane, and the nucleus there, okay? So those two things, what we need for regeneration to occur is that the cell body's intact, and if we have enough neural lemma, all right, that we can work with to help regenerate, all right, the axon. Now, okay, trauma can vary with minor trauma to severe trauma. So, all right, we're more likely to have a successful regeneration, all right, if there's less damage. Well, that makes sense, all right? And if the distance from the site of damage to the structure to which that axon innervates is shorter, okay? So that means if the structure, that effector is either a muscle or a gland, all right, the closer that damage is to that muscle or gland, all right, the more likely, all right, you'll have a successful regeneration. So let's just walk through the steps of regeneration. All right, so the first step, let's sever the axon. Okay, that's what happens here. That's what we're seeing up here. Okay, we've severed the axon. Okay, so basically what's going to happen is everything downstream towards our effector, and in this case, it's going to be skeletal muscle, right? All that starts to degenerate. That axon starts to degenerate, all right? But we have some of that neural lemma left over, okay? So that's what we're going to see in step one. Trauma occurs. Now, in step two, there's two things that are going to happen. Proximal to the cut, okay? That's going to be closest to the cell body. The axon is going to seal itself off. Makes sense. You know, it's like if a ship is taking on water, you got to seal off the holds there, right? It will seal itself off, and then that end will swell up. Almost it'll look like a synaptic knob kind of swelling. Now, distal to the cut, everything starts to degenerate. Okay, we call that Wallerian degeneration. All right, but that neural lemma survives, and that's going to be the important aspect. We need that neural lemma, all right, to survive, because we're going to kind of thread that axon back through that neural lemma there. Okay, so whatever's left over, All right, that neural lemma and the endoneurium. What's the endoneurium? The endoneurium is that connective tissue sheath that covers each individual axon. So we have the neural lemma and the endoneurium. And so those two create what we call the regeneration tube. Okay, the regeneration tube. And that's going to be that tube that's going to prompt and help that axon to grow. So let's see the picture here so far. Okay, so here's our damaged area. Now we notice that proximal to that, towards the cell body, okay, the axon has sealed itself off and it's swollen. Everything downstream starts to degenerate. The neural lemma stays and the endoneurium stays. But everything else seems to just go away. So what's left behind is that neural lemma and the endoneurium will form this regeneration tube. And so what this regeneration tube is going to do is it's going to start to produce these chemicals. Okay? And they're called nerve growth factors. And so what will happen is, all right, these nerve growth factors are going to promote the axon. It's like a dog when you try to get it to come over towards you. If you have a treat, all right, they can smell that treat and they come over, all right? Well, these nerve growth factors are going to kind of stimulate the axon to move in the direction, all right, towards whatever our effector is. So that's what we'll see here. Whoops, where am I going? Boo, hold on. Sorry about that. All right, so that's what we're seeing. You can, and if you look here at the picture, okay, here's where our axon is, right, and you can see it starts to just get closer and closer to the effector. And that's what will happen. These nerve growth factors are going to promote the axon to move closer and closer and then eventually reinnervate that tissue there. Pretty cool. Okay. Then eventually we'll start to innervate that effector again, and all is well. Okay. What happens in the central nervous system? Okay. Well, unfortunately, regeneration is extremely limited. It will not happen as easily as you saw in the peripheral nervous system. And the part that kills me, and I need to do more research into this, but you noticed how the peripheral nervous system, the neurolemmacytes, the glial cells that help to myelinate the cells, produce that growth factor. Well, the oligodendrocytes secrete growth inhibiting molecules. Okay, inhibiting, not growth factors, but inhibiting molecules. So what that does is that is going to prevent those neurons, those axons. And so it obstructs that growth there. What happens? Remember what we said? One of the roles of the glial cells of the astrocytes is to fill in spaces in which neurons are dying. Well, that's what's going on here. Those neurons are dying, so those astrocytes slide on in and form scar tissue and actually prevent, all right, the regeneration of the central nervous system. It's unfortunate it does that, but that's just how it is. All right, let's do some labeling. We'll do some labeling. All right, so if you have your atlas with you, why don't you go ahead and pull that out? There's only 17 slides to label. So unfortunately, I do not have a Quizlet for this. Okay, I do not have a Quizlet. But I do have a Quizlet for everything else. That's good news. Oh, come on. My cursor's gone. It disappeared on me. That was weird. Okay, I don't know what was going on there. You guys probably thought I was crazy. Oh, wait, you can't see anything. That's why. All right, sorry about that. Oh, my lordy, lordy. All right, let's try this again. All right, now you should be able to see this. Okay, so let's start off with the neuron here, and we're going to start off at the receptive segment. The receptive segment includes the cell body, which is known as the soma, and the dendrites. So right here, what you're looking at in the circle there, that is the cell body or the soma. Okay, in cell bodies, we're going to have a lot of the organelles, the nucleus, the nucleolus. That's where we're going to see, all right, this substance here. All right, there's the nucleus. Okay, to me, it looks like a clear golf ball. Okay, here's one of our organelles, but we're seeing the mitochondria down here into the axon, which is good. All right, and I'll talk more about this in lecture, right? But neurons have tons of mitochondria because they got lots of work going on with what they have to do. So it's important that we have the powerhouse of the cell, and you want to have that to be in abundant numbers there, right, in the axon there. So that's the mitochondria, right? Here you can see on those blue tube-like structures and the endoplasmic reticulum, that arrow should be pointing at those little red dots there. That's the Nissl bodies, or the chromatophilic substance. It's up to you which term you want to use. I know which one I'd be using, okay? Nissl is a whole heck of a lot harder to misspell than chromatophilic, at least for me. Okay, and so that is going to be, those Nissl bodies are those ribosomes. All right, that arrow is pointing to a dendrite, one of those short processes that come off of the cell body. Remember what their job is: to receive information or input and transmit it to the cell body. And you'll see why that is when we start talking about graded potentials. But there's our axon. There's our conductive segments, right? That is going to conduct, all right, our action potential from the cell body end of the cell all the way down to the terminal end of the cell by the synaptic knobs. Okay, so that arrow is pointing, and that think of the axon like your cell phone charging cord. Okay, it's the entirety of the cord. The entirety of the cord. So look, the arrow did not move between this slide and this slide. Look, there's the axon and the axolemma. Okay, the axolemma, all right, that arrow is pointing just to the white covering there. Okay, if you look at the inside of the axon, you got all those little structures in there, the mitochondria, the neurofibrils, all right, but also you'll have the axoplasm, okay? But this arrow is pointing to the white covering. That is the axolemma. That is the name of the plasma membrane of the axon. Then at the end of each axon, you will have the telodendra, or the axon terminal. Basically, that's where the axon splits off into these little divisions here. And at the end of these little divisions are going to be those swellings, okay? So the telodendra are these guys right here. At the end of the telodendra are those structures, the synaptic knob or the synaptic end knob. And that's going to be filled with the synaptic vesicles, and the synaptic vesicles will contain our neurotransmitter. And then our neurotransmitter will be released onto another neuron or an effector. Okay, here is our axon hillock. This is also known physiologically or functionally as the initial segment of the neuron. So you have the receptive segment, which is, all right, the cell body and the dendrites. Then you have the initial segment, which is the axon hillock. Then you have the conductive segment, which is the axon. And then our final segment is the transmissive segment, and that's the synaptic knob. Okay, so there's our axon hillock. Again, almost guaranteed that you might see it on a test question. All right, the axon hillock is the area in which we initiate or generate an action potential. All right, and then covering the cell here of our neuron is our neurolemmacyte, or our Schwann cell. So the entirety of this cell will wrap itself around the axon in the peripheral nervous system. And it'll create that myelin coat, or that myelin sheath. So you can see there's the Schwann cell nucleus pushed up to the edge of the on the periphery of the neurolemmacyte. And then you can see that the arrow is pointing to the myelin sheath. So you might get confused here. Look at this. Here's the myelin sheath, and then the arrow is pointing to the Schwann cell. So on a test, if the arrow is pointing directly here, and there's no words whatsoever, this is your correct answer. Myelin or myelin sheath. If it's pointing to the same area and it says name the cell, then you name the cell, and this is your correct answer. Schwann cell, neurolemmacyte. All right, so just keep that all in mind. Like you don't have enough to keep in your mind, but still. Okay, and then you'll notice, all right, where this bracket is from one end of